Right, I think we'll move on to the next speaker. Oncologists, whether they are surgical, radiation, or medical all over the world, they face with the eternal problem of catching lung cancers late in the natural history of the disease. And if you were to actually look for them and identify at an early stage, the mortality rates will be much better. But is that so simple, or is there a diversity which is genetically based amongst populations? Is what Dr. Brid Ryan from the prestigious Maryland, USA National Cancer Institute is going to talk to us about. She is a Statman investigator at the National Cancer Center. She was among the first to demonstrate asymmetric division of the DNA in cancer. Her lab oversees an integrative and translational approach to lung cancer research, examining the genetic, environmental, and biological contributions to racial disparities in lung cancer incidence. Her research program also develops biomarkers for early detection of lung cancer, and our topic is going to be population diversity in lung tumor biology in the United States. Over to you, Dr. Ryan.
Thank you very much. Right, raise your hand if halfway during the talk you either don't understand me. I'm from Ireland, so sometimes people find it hard to understand, or if the volume goes, just raise your hand. So I want to begin by saying a very sincere thank you to Keshav and the organisers for inviting me to come and speak here today. It's my first time in India, and it's been a wonderful experience so far, and I'm very, very much enjoying the meeting. I work at the National Cancer Institute in the United States, so we always have to start our talks with this disclosure, and just to say that the data that I present today and the views that I express are my own and not those of the US government. And as it pertains to the work that I'm going to present today, I don't have any conflicts of interest to disclose. So as we mentioned, the main focus of our laboratory is lung cancer. Now we know lung cancer today to be the leading cause of cancer-related death both in the United States and worldwide. But at the turn of the 19th century, lung cancer was actually quite a rare disease. And I often start with this because I think it is important to appreciate the rapid rise in the public health burden that occurred in the 1900s. In 1940, lung cancer became the second leading cause of cancer-related death. And there was two very important studies, well, several quite important studies around the 1950s and 1954. So it began with case control studies and then a cohort study. And these really they were done in the United States and they were done in England. And they were the first studies to really scientifically and quite conclusively tie tobacco exposure with lung cancer, because of there are different causes, and we'll talk about that in a little bit. But without question, smoking is the single largest etiological factor associated with lung cancer development. And in 1987, lung cancer became the leading cause of cancer-related death, and unfortunately that remains the case today. So in the United States and around the world, we've been tracking lung cancer incidence for many years now. And I've put an arrow here at around 1972, because in the United States, it was the first time that they actually started to track lung cancer incidence by racial and ethnic group in the United States. And these were two seminal papers that showed in the same year the realization that the incidence of lung cancer was not uniform across racial and ethnic groups in the United States. And there was this, as they called it at the time, a non-white dominance, which we now know is linked primarily in African Americans. So what I've graphed here, of course, at the very beginning of those studies, they just looked at differences in lung cancer incidence between European Americans and African Americans. But now, over the years, we have a more detailed breakdown of racial and ethnic groups within the United States. And what I hope you can see here is African Americans shown here in black. And compared with all other racial and ethnic groups, the incidence of lung cancer is higher in this population, as is mortality. The important thing to note about this graph, and something that still continues to give us pause for thought when we're thinking about our research, is that this increased incidence of lung cancer is restricted to men. We don't see the same trend in women, and this is something that we still don't fully understand. As I'd mentioned, tobacco is the major factor associated with lung cancer development. It accounts for about over 80% of all lung cancer cases. This is a nice graph that shows how the rise in smoking exposure really did mirror the rise in lung cancer that occurred later on. And I always put up this graph because I think from a public health perspective it's extremely important, because it shows that no matter at what age you stop smoking, your risk of lung cancer will start to decline. So I think it's always important to make that point as well. But because tobacco is the major factor associated with lung cancer incidence, when people sat down and looked at lung cancer disparities within the United States, obviously tobacco is one of the first things that they thought of. However, by and large, the incidence of lung cancer that we see in African Americans is inconsistent with tobacco exposure, and we can break that down in various ways. The first thing is that we know, just for many other types of cancer as well, African Americans actually have an earlier age at diagnosis. They have a lower overall tobacco burden. They smoke fewer cigarettes per day. And even if you compare the risk of lung cancer controlling for the number of cigarettes per day, African Americans still have a higher lung cancer incidence. And this is work that was done initially in 2006 by Chris Hyman and Loic Lamarchand, but it's been reproduced several times over. African Americans also start smoking later in life, so the cumulative period of exposure is shorter, yet there's a higher incidence. And within the United States, one of the smoking behaviors that's particularly different is that they primarily use menthol cigarettes, and there is no evidence that the relative risk of a menthol cigarette is higher than that of a non-menthol cigarette. In fact, it seems to be the opposite. So in as much as that we can understand tobacco exposure, we do not understand how it does or does not contribute to disparities. There are differences or racial and ethnic differences in cancer survival as well. I won't go over them in depth in this talk, but just to say that differences in survival do seem to be linked to access to care and access to high quality health care within the United States. So there are two key focuses and research questions within our lab. The first is trying to understand why the incidence of lung cancer is so much higher in African Americans, and especially African American men. And the second is to ask whether or not there are any biological differences in lung tumors from European Americans and African Americans, and if so, can we leverage those differences to improve patient outcomes? We're a mixed methods laboratory, so we use a lot of descriptive epidemiology and molecular epidemiology to answer this first question. But the focus of my talk today is going to be on question number 2, and for that we use a little molecular epi, but also primarily integrative tumor biology and precision medicine, so a lot of omics and genomics. So the main workhorse for our studies is the lung cancer case control study. It was started in 1998 by Kurt Harris, and this is located here in Baltimore. One of the advantages of this study for doing disparities research is that it's overrepresented with African Americans. We collect blood and tissue when it's available if patients go to surgery. But importantly, we also collect very detailed questionnaire data, and this really is a very important and novel aspect of our study. I wanted to show this because it gets back to something that was mentioned in the talks yesterday. So when patients are recruited into our study, they self-report as either African American or European American. But that is by and large, of course, a social construct, and we know that the underlying genetic diversity and admixture within those populations can be quite different. So what's shown here is a study that we did a couple of years ago where we actually went back and measured the genetic diversity in each of the participants in our study. And as you can see, among African Americans, shown here in purple is West African ancestry, so you can see that primarily the median African ancestry is about 75% in these populations, where in the European Americans you can see they are predominantly of European ancestry. And so these are factors that we take into account as well in our genomic studies. So one of the first studies that we did, continuing the theme of the last talk, was we looked at the relationship between circulating immune and inflammation proteins with lung cancer risk. So again, this is looking at the blood, but a specific aspect of tumor biology within the circulation. We went to the case control study and we selected healthy individuals and also individuals with lung cancer. And we basically measured 30 immune and inflammation proteins using a very sensitive ELISA assay. The first study that we did was in European Americans. For this, we actually used the NLST study, which some of you may be familiar with as a study which led to the introduction of lung cancer screening within the United States. And we used 265 case samples and 530 controls. When we did this, shown here in red, you can see the proteins that we found to be positively associated with lung cancer risk: IL-6, IL-8, CRP, and interferon gamma. We then went to do a similar study, as I mentioned, in African Americans, measured the same proteins. But as you can see, there's many more proteins associated with risk than in European Americans. These same 4 proteins are found, but there are many others as well. Now within our lab, replication and validation is always very, very important. So we collaborated with a very good colleague of mine, Anne Schwartz, who works in Detroit at the Wayne State University. And in her study, we again took another 400 controls and 400 cases, and they were nicely matched for smoking history. And we were able to validate, 11 of those 13 associations. The 2 that didn't actually, so with Anne's study, we looked at plasma instead of serum. And in her study, the 2 that didn't validate actually have quite low levels in plasma. But this validation aspect was a very important part of the study. So when we line up the European Americans against African Americans, what I hope you can appreciate is that the circulating immune and inflammation profile between these 2 populations at the time of a lung cancer diagnosis is actually quite different. And so within the lab, of course, we're trying to think to ourselves, well, what does this mean? How can we understand these data further? One lens that we have in our lab is always translational, so thinking forward, how can we use these data to improve patient outcomes? And one of those, of course, includes biomarker development, and this was mentioned at the beginning of the introduction. How can we detect cancer earlier? The study that we did detected samples, or took samples from patients at the time of diagnosis. But of course, to be really useful as a blood-based biomarker, we need to be able to detect samples, or to detect individuals at risk of lung cancer before it actually becomes clinically relevant. And so we're now working with colleagues at Vanderbilt with the Southern Community Cohort Study, which is a prospective study, just like the one that was talked about a moment ago, where we follow people over time, and also the PLCO study, which is a screening study run by NCI. And with these 2 studies, we're working with all of the proteins that we'd found in our study, we're measuring them and asking the question, are they actually associated with lung cancer risk? The second question, of course, is, well, why do we see this difference in the first place? We thought it might be menthol cigarette exposure, because that is one of the key smoking behaviors that I mentioned was different, but it absolutely isn't in the data that we have. So, of course, then it became possible that there was differences in immune cells within the tumor tissue itself. Well, when we looked at the proteins that were similar and the ones that were different between both populations, many of the proteins that we only found associated in African Americans seemed to be related to macrophage biology. So we went back to our case control study, and where we had samples from patients that went to surgery, we started to measure levels of macrophages. So initially with this study, we've just used a pan-macrophage marker, CD68, and our data suggests that using this marker, there is no difference in CD68 infiltration in European Americans or African Americans. But of course, we know, and many of you in the audience will know as well, that a macrophage is not a macrophage. It could be an M1, it can be an M2, there's even subsets of M2. It's actually quite complicated. And so what we've now gone back and are in the process of doing is doing multiplex IF, where we have specific markers that within a single section can identify each of the individual subtypes. And of course, this is also important because while M1 macrophages can actually be quite pro-inflammatory, M2 can be more immunosuppressive. So it is important to differentiate them, and this is work that's ongoing. And another work that I won't talk about today, of course, is the other lens that we have is we're thinking about, well, why do these differences, what is driving them, and also can it give us clues to the etiology? And so in the lab, we're currently doing some of these studies as well at present, and maybe in question time we can talk about some of that. The second part that I want to talk about is really a lot of the precision medicine type of studies that I had mentioned to you. And to us, understanding cancer at the molecular level is extremely important because most of the new treatment strategies that are coming downstream, both for at the first incidence of cancer, but also when recurrence happens, is relying heavily on the molecular features of a tumor. And most of the work that has been done to date in the United States has been focused on populations of European ancestry, with quite a low focus on including tumors from other minority populations. And so what we've been doing for the past couple of years is mining our database of tumors from both European Americans and African Americans, but primarily African Americans, and looking at the various layers of the genome, looking at obviously including demographics and exposures, but also looking at DNA, RNA, and methylation. Now obviously I won't go through all of those today, but I do want to, in the last 8 minutes or so of the talk, go through with you some of our findings from looking at the DNA layer of the genome. So many of you will be familiar with a graph that looks like this. This one is taken from TCGA. And what it shows essentially is a somatic breakdown of mutations that are found in tumors primarily from European descent. And many of these genes will be familiar to you. So from the work that has been done previously, we know that there is one key difference between European populations and Asian populations in lung cancer genetics, specifically among never smokers, and that is the presence of EGFR mutations, which are really predominant in Asian populations, and especially, as I mentioned, in ever smokers. So in our study, when we included African Americans, I just want to show this again. For the samples that I'm going to show you, this is the genetic admixture for those populations with, again, as you can see, a predominance of African ancestry and a minimal proportion of European ancestry in the patients. Everybody has self-identified as African American, but you can see there is a difference in the proportion of the African genome within these patients. So the first study that we did was done on fresh frozen tissue. It was matched tumor and normal, and we looked at a targeted panel of about 560 genes. The first message of this and the first results that I want to share with you is that by and large, the frequency of the key driver genes that I showed you in that first TCGA picture, they were primarily the same. So when we got the first batch of results, there was nothing that jumped out as telling us the somatic mutation profile is extremely different. It's not. But when we, and this is just an example of what it looks like. But when we looked a little bit further and we looked at the tables and the frequency of mutations, we noticed that the frequency of two specific genes, JAK2 and PTPRT, were certainly higher amongst African Americans. Again, validation is a theme that's very important in our lab. So before we actually hang our hat on something and necessarily believe it, we went and tried to get access to other populations and other data. And in fact, we validated our observation. Essentially, mutations in PTPRT are found in about 20% of lung adenocarcinomas, whereas they're found in about 8% of European Americans. And mutations in JAK2 are found in about 10% of African Americans and just about 2% of European Americans. And combined, because these genes are primarily, the mutations, I should say, are mutually exclusive, what we have is about 30% of patients that carry mutations in these two genes in African Americans and about 10% in European Americans. So you might wonder, why do I care so much about these two genes? Well, I do now, because when we looked at them, what we realized is that these two proteins and genes actually function within the same pathway. And it's the JAK-STAT pathway downstream of IL-6. So PTPRT is a protein phosphatase that inhibits the phosphorylation of JAK3, while JAK2 is actually a factor that stimulates phosphorylation of STAT3. Just because you have a mutation in a gene doesn't mean necessarily that it's going to be active or be meaningful in any way. And so what we did was we integrated RNA-seq data that we had for these patients and classified them as either mutant or wild type for these proteins, and then asked was there any enrichment in particular pathways. And what we saw was that there was enrichment in IL-6 JAK2 pathway, interferon gamma pathways, so all pathways that were downstream of here, which gave us confidence that this was in fact evidence for overactivation of this pathway among patients that had these mutations. Downstream of this pathway, STAT3 is a microRNA called miR21, which is very near and dear to my mentor. So anytime it comes up, we always have to look at it. But similar to what we did here, we measured expression of miR21 and miR181B. These are both downstream targets of this pathway, and they're oncogenic miRs, especially in lung cancer. And what we find is that expression of these two microRNAs also is elevated amongst mutated samples, which is more evidence for hyperactivation of this pathway amongst the mutations. And so what this does then is it gives us confidence that perhaps this is a new pathway that is potentially actionable and targetable for these patients. And so what we're currently doing in collaboration with a very good friend and colleague of mine, Sharon Pine at Rutgers University in New Jersey, is taking patient samples from individuals that either have the mutation or don't. And this pathway is very tractable for us because there are many, many JAK2 inhibitors and STAT3 inhibitors that are already FDA approved and in clinical trials for other conditions. And so what we're currently asking is whether or not patients' tumors that carry these mutations, whether or not they'll be more likely to respond to these drugs. And that work is ongoing. So in the last part of my talk, I just want to mention another recent findings that we actually just published this week. And again, the same way, we looked at the somatic genome, but this time instead of looking at mutations, we looked at copy number. The first thing, of course, that you do is, you look to the driver mutations and the genes that we know are important in lung cancer. And just to show here on this straight line, any gene that falls on the line has a similar frequency between European Americans and African Americans. And if it falls off the line, then it's more likely to be found in just one population. And essentially what we found is there was actually much higher amplification of KRAS in African Americans that correlated with expression. And there was actually a lower deletion of CDKN2A and PTEN, which again are two important tumor suppressors. We also looked a little bit more broadly, and we looked at all the focal regions in the genome. And this is quite a complicated picture, but there's two key points I want to share with you from it. The first of those is that when you compare all the somatic copy number gains and amplifications and losses between these two populations, what you find is that about 60% and 70% is shared. That actually means that a lot is not shared and that a lot is specific to each population. And the other thing that we found is that the pattern of co-occurrence of these various alterations, as you can see, if you compare from left to right, it's very different between these populations. And what that suggested to us is that genome-wide, at the somatic copy number level, these genomes didn't look very similar. So we went back and we quantified this in a slightly different way. And we used the data to get a measure of genomic instability. Genomic instability is I'm sure, a very common theme to you, but in the way that we measured it was just basically asking in each population how much of your genome was messed up or perturbed. What we found is that in lung squamous carcinomas, but not lung adenocarcinomas, there was a higher incidence of genomic instability and homologous recombination repair deficiency, and also these complex genome rearrangements called chromathripsis in African Americans. But we didn't see this in squamous, we only saw it in adeno. And we used TCGA to validate these observations. Of course, again, we're wondering, well, why is that the case? Why would we have more homologous recombination repair deficiency in African Americans but not European Americans, and why is it only in squamous? Well, I can tell you we don't know the full answer. But one thing that occurred to us was that perhaps it could be driven by the germline. So we leveraged a big database that TCGA had of about 10,000 individuals where they had sequenced the germline DNA and then catalogued all the variants that they had as either pathogenic or normal. And we looked at all the pathogenic variants that we could find and compared their frequency between European Americans and African Americans. And what we found is that both pan-cancer and also especially in lung squamous cell carcinoma, there was a much higher frequency of these germline homologous recombination repair deficiency genes in African Americans, whereas in adenocarcinoma it was equal. This doesn't categorically tell us that the origins of what we're seeing in somatic tumors is driven by germline, but it certainly is an indication that it might be. And so we're going back now to some larger databases where we have access to more germline information to try and verify this further. In the last slide I just want to show you is that what we saw in lung cancer with a higher prevalence of homologous recombination repair deficiency in African Americans is actually seen in many cancer types. So when we looked at TCGA at 17 of the 23 cancer types, what we actually see is that there is also a high prevalence of homologous recombination repair deficiency and genomic instability and these complex genome rearrangements found in African Americans. And this was interesting to us, of course, because it also was mirrored a paper in Nature Communications last year that showed, again, for breast cancer, especially triple negative breast cancer and from women from Africa, there was again a higher prevalence of genomic instability and homologous recombination repair deficiency. So these are basic biology findings, but why does it matter? And is there any way, going back to the beginning of the talk, that we can leverage this in any way to improve patient outcomes? Well, in a very similar design to what we're doing earlier, many of you might know that the prevalence of homologous recombination repair deficiency, which is what happens when you have BRCA loss, makes those tumors incredibly susceptible to PARP inhibitors. And in fact, PARP inhibitors are now first-line therapy for many cases of breast cancer, but also ovarian cancer just recently. And so it raises a very simple question. For tumors that currently you would never consider using a PARP inhibitor because we don't consider them to be BRCA mutant or to have high levels of HRD. Well, what if you did measure HRD and you saw high levels? Would those tumors now become targets for PARP inhibitors? And that's exactly what we're doing now in the lab. We have taken cell lines from European Americans, African Americans, and we've essentially measured HRD in all of those samples, and now essentially asking the question, does a higher level of HRD within lung cancers sensitize those cells for PARP inhibition and PARP therapy? I didn't share it with you today, I only shared you the stories from here, but obviously we've also looked at the transcriptome, the methylome, and we're currently doing studies to integrate all of these. The work isn't completed, it's still ongoing, but we find as we go through it, the more we look for differences, the more that we actually do find them. So in summary, I just want to say I focused obviously on what was different, but I want to leave you with a very important message that a large proportion of the tumor biology that we have looked at is shared between European Americans and African Americans, and I do think that's an important point. We do, of course, find some differences. We think some of these could be translationable or have potential for translation, but a lot of the biology is shared, and because smoking is the single major cause of lung cancer within the United States, that's not a particularly unusual finding, but yet once we look, we are able to see differences. Our research lens, as I hope I've showed you, is bidirectional. It's translational. We look forward and think, how can we translate the differences that we see into hopefully new types of therapy? But it's also reflective. So when we have our findings, we try and look backwards and ask ourselves, can it give us clues to why we see these differences? And of course, that relates to the first question that I told you about, is as yet we really don't understand why we see these differences. So with that, I'd like to finish. I had an acknowledgement slide, but it seems to have disappeared on me.